So, as a research technician, what do you do here at Stroud?
Here at Stroud, I get to do two different kinds of work. Part of what I do is in the lab, where I work with what we call molecular techniques, which is looking at DNA, using DNA to study our environment. The other thing that I get to do is actually go outside, get hands-on in the stream, get wet, catch fish, dig up mussels, all kinds of that really fun, dirty stuff.
So what is DNA? Can you tell us that real quick? I think
Sure.
the students have a background, but just remind us.
DNA stands for deoxyribonucleic acid, which is a big word, but is a very, very tiny thing. And it's the building block of life. So in your cells, the DNA is what controls the cell, what makes the proteins, what drives all the processes of life.
Well, and it's made up of how many bases?
There's 4 different base pairs, A and T, and G and C, and those bind together and form the DNA double helix.
Perfect. So is there a way that we could actually see DNA in action, see it?
Since it's very small, you can't look at it directly unless you have a very, very powerful microscope. But what we can do in the lab is visualize it using gel electrophoresis. And the way that works is we make a gel, which is made of agarose, and it basically looks like clear jello. So it's flexible and kind of rubbery. And we take our DNA sample and we pipette it into one of these little wells.
Ooh, that's tiny.
And then when we apply an electrical current to that, the DNA will move through the gel. And the smaller the piece of DNA is, the farther it'll move. So what we can do is look for a target piece of DNA that we know the size of. And so this is what a gel will look like when it's done. This is our ladder, and these are known sizes of DNA.
So the top are the smallest or the biggest?
The biggest. So this is 10,000 base pairs, this is 5,000, all the way down to 100 base pairs. And then this is the target DNA that we're looking for. So what this gel tells us is that all of these samples have the piece of DNA that we want, and that these samples, where it's completely absent, it's missing from there.
Perfect. Well, thank you so much for your time.
You're welcome. Enjoy your gel pouring.